Nucleic acids. Nucleic acids are components of all living cells, which are responsible for the storage, transmission, and translation of genetic information for the synthesis of proteins. Nucleic acids are mostly in the form of nucleoproteins, a complex of basic proteins and nucleic acids. There are two types of nucleic acids, namely deoxyribonucleic acid or DNA and ribonucleic acid or RNA. Buret's test is used to detect the presence of peptide bonds. In this test, potassium-sodium tartrate is added to chelate and thus stabilize the cupric ions in the solution or to maintain their solubility in an alkaline solution. The reagents used are copper sulfate and sodium hydroxide. A positive reaction would yield a pink or purple color, and a negative reaction would yield a blue color. The 10% of ammonium hydroxide and 2% of silver nitrate is a test to detect the presence of a purine base. The hydrolysis of N-beta-glycosidic bonds between purine bases and ribose or deoxyribose results in release of purine base. The reagents used are silver nitrate and ammonium hydroxide. A positive reaction would yield a gelatinous white precipitate. Wheeler-Johnson's test is used to detect pyrimidines, uracil, and cytosine. This test is based on the action of bromine on either of these two pyrimidines in aqueous solution. The reagents used are bromine and barium hydroxide. A positive reaction would yield a green color with bromine or C and U turns into dialuric acid, and purple color solution with purple precipitate for barium hydroxide. Murexide test is used to detect the presence of uric acid and some other purines. The reagents are nitric acid and dilute ammonia solution. A positive reaction would yield a reddish violet color for ammonium purpurate and pink color for purine alkaloids. Bial test is used to detect the presence of pentose sugars through a process called condensation. The reagents used are arsenol, concentrated hydrochloric acid, and ferric chloride. A positive reaction would yield a blue-green color or ribose sugar, and a negative reaction generally gives green, red, or brown color for hexose sugar or such as glucose and fructose. This test will detect the deoxyribose of DNA and will not interact with the ribose. The reagents used are concentrated sulfuric acid, aniline, diphenylamine, and phosphoric acid in acetone or ethanol. A positive reaction would result in a blue color intensity that is proportional to the amount of DNA found in the sample, and green color indicates the presence of RNA found in the sample. A negative reaction gives off a clear color. Ammonium molybdate test determines the presence of a phosphate group by undergoing hydrolysis of pyrophosphate to phosphate. Sulfuric acid is the reagent used. Yellow precipitate is seen in a positive reaction and white solution is seen in a negative reaction.